Good morning, my dear student. On the behalf of KTGM College, Nasik and Department of Chemistry of this college, I am Dr. S.R. Labhade. Welcomes you to the lecture on polarographic methods of analysis. Dear student, polarographic methods of analysis, that is a technique of voltammetric analysis. Voltammetry voltammetry comprises a group of electroanalytical technique in which current, voltage, time, relationship during the electrolysis of sample is utilized for qualitative and quantitative analysis of the sample. Voltammetry, that is a group of electroanalytical technique in which current voltage time relationship during the electrolysis of sample is utilized for qualitative and quantitative analysis of sample. Here, voltammetry is an electroanalytical technique. Electroanalytical technique are those technique in which electrode system is utilized for determination of electrical property of solution, electrical properties of solution or sample solution. In a voltammetry, electrode probe are inserted into analyte solution and electrical property of solutions are determined for qualitative and quantitative analysis of sample. In voltammetry, analysis is carried out. Qualitative and quantitative analysis in voltammetry is carried out by using current voltage time relationship. That means a graph of current as a function of voltage or a graph of current as a function of time at a constant voltage, and therefore here current voltage time relationship during the electrolysis of sample is utilized for qualitative and quantitative analysis in voltammetric method. As we discuss here, it is an electroanalytical technique. Therefore, electrode system employed in voltammetric work consisting of 3 electrodes. First electrode is called as working electrode. Working electrode is very small electrode with very small dimension. Electrode with very small dimensions and therefore it is also called as microelectrode. and on this electrode the analyte reaction takes place. Therefore, it is called as a working electrode. Analyte reaction occurs on a working electrode. Therefore, first electrode in voltammetric work is a working electrode. Dimension of working electrode is very small. Therefore, it is also termed as a micro electrode. This micro electrode or working electrode is get polarized with the application of very small potential difference. Because of its very small dimension, current density at working electrode is very high. Current density means it is a ratio of applied current upon area of electrode. As area of electrode is very small, current density is very large, and due to this, working electrode or this small size electrode is get easily polarized, and therefore analytes reactions taking place on the working electrode. Second electrode, here this working electrode carries the electrolysis current. Working electrode carries the electrolysis current. Electrolysis current. Second electrode is known as auxiliary electrode. Auxiliary electrode. Auxiliary electrode, it is used in combination with the working electrode. Used in combination with working electrode, and it also carries electrolysis current. But dimension of this auxiliary electrode is very large compared to working electrode. Therefore, polarization of this electrode is not possible, and no analyte reaction taking place on auxiliary electrode. If we have to measure the potential of working electrode, or if we have to control the potential of working electrode, then third electrode employed in voltammetric work is called as a reference electrode. Reference electrode such as saturated calamine electrode. Generally, saturated calamine electrode is used as a reference electrode in all types of voltammetric work. Therefore, reference electrode is used to measure the potential of working electrode. Therefore, it is coupled with the working electrode to monitor or to control the voltage of the working electrode. Students, now we discuss the working of voltammetry, how voltammetric analysis is carried out to determine the analyte concentration and to identify the analyte. In voltammetric analysis, there is a polarographic cell. In this polarographic cell, 3 electrodes are inserted. First electrode is called as auxiliary electrode. Second electrode is working electrode. Working electrode. Third electrode is reference electrode. Now these 2 electrodes are coupled by using a voltmeter in order to control the potential of working electrode. Here a analyte solution is placed. Analyte solution. Now here these 2 electrodes are connected to a power supply through variable resistor in order to apply a variable potential. Now this is a negative port, positive port. This is a sliding contact C. Now this is a diagram that represents voltammetry, and in voltammetry current is measured, therefore here is galvanometer. Voltammetric analysis is carried out by using a electrolysis cell consisting of 3 electrodes. One is working electrode, second is reference electrode, and third is auxiliary electrode. All 3 electrodes are inserted into assembly containing analyte solution. With application of variable potential, here working electrode across the working electrode and auxiliary electrode, electrolysis of this analyte solution is carried out. Now we discuss how this voltammetry work or principle of voltammetric analysis. Voltammetry principle. In voltammetric analysis, an analyte solution is made a part of electrolysis cell containing polarized working electrode and non-polarized auxiliary electrode. Therefore, analyte solution is placed into electrolysis cell containing working electrode and auxiliary electrode. With linearly increased potential, analyte solution is electrolyzed. Analyte solution is electrolyzed when analyte undergo oxidation or reduction at working electrode. That potential is the identity of the analyte. The current is measured, which is directly proportional to during redox reaction. The current is measured and is directly proportional to the concentration of the analyte. In this way, voltammetry work. Therefore, in voltammetric analysis, analyte solution is made a part of electrochemical cell or electrolysis cell containing working electrode and auxiliary electrode. with linearly increasing potential of working electrode and auxiliary electrode, analyte solution is electrolyzed. The applied potential acts as a driving force for the redox reaction. The potential at which the analyte undergo oxidation or reduction is its identity, and during oxidation or reduction, magnitude of current is directly proportional to concentration of the analyte. Therefore, both qualitative as well as quantitative analysis is done in a voltammetric analysis. Student, voltammetric work is carried out by two different ways. In first way, we can measure the current as a function of linearly increasing potential. Therefore, voltammetry may be performed by number one, measurement of current as a function of linearly increasing potential. Or second type, current is measured as a function of time at a constant potential. First type is measurement of current as a function of linearly increased potential, or second one is measurement of current as a function of time at a constant potential. Here, voltammetric method particularly measures the concentration of analyte solution in terms of the current measured during the oxidation or reduction of the analyte using this electrolysis cell. When current is measured as a function of potential, generally those are termed as a polarographic analysis or hydrodynamic voltammetry. Sometimes current is measured as a function of time at a specific potential value. Those are termed as a potential program voltammetry. That's the classification of voltammetry. Voltammetry, no doubt, it is an electroanalytical technique in which current voltage time relationship, current voltage and time relationship during the electrolysis of analyte is utilized for qualitative and quantitative analysis of analyte. Therefore, voltammetry are classified on the basis of on the basis of the voltage swept program. That means how voltage is applied to working electrode on the basis of voltammetric techniques are classified, and first technique of voltammetric analysis is called as linear scan voltammeter. First technique of voltammetry is called as linear scan voltammetry. In this voltammetry technique, potential is linearly scanned. Potential of working electrode of working electrode is linearly scanned and a current is measured. And therefore it is called as a potential scan or linear scan voltammetry. In linear scan voltammetry, the potential of working electrode is linearly increased. The potential at which analyte undergo oxidation or reduction is its identity, and at that potential current is measured. The measured current is directly proportional to concentration of analyte, and potential is the identity of that material. This type of voltammetry is called as linear scan voltammetry, and linear scan voltammetry classified under two categories. First one is called as polarography. Polarography is a linear scan voltammetry, but here working electrode is a dropping mercury electrode. Working electrode is made up of mercury, and second type of voltammetric work is called as hydrodynamic voltammetry, in which working electrode is made up of a stationary electrode like platinum stationary electrode, like platinum, gold, silver or graphite like this. Therefore, in linear scan voltammetry, the potential of working electrode is linearly increased and current is measured. Here, this linear scan voltammetry, when it is performed by using mercury as a working electrode, that technique is called as polarography. And when this linear scan voltammetry is performed by using any other stationary electrode such as platinum, gold, silver, graphite, etc., then it is known as hydrodynamic voltammetry. Another main difference between these two techniques is that in polarography, analyte solution is quiescent, it is unstirred. But in hydrodynamic voltammetry, analyte solution is continuously in motion. Therefore, it is carried out by using a stationary electrode, and here analyte solution is quiescent, and therefore we can carry out that analysis by means of mercury electrode. Now, second type of voltammetric technique is called as potential program voltammetry. Potential program voltammetry. In this case, potential of working electrode is programmed, that is not linearly increase or decrease, but it is increase or decrease according to requirement of the reaction. Therefore, it is called as potential program voltammetry. Potential program voltammetry are classified into four different types. Number one is potential step voltammetry. Potential step voltammetry. In potential step voltammetry, current is measured after application of potential pulse to working electrode. Potential pulse to working electrode, and therefore it is called as potential step voltammetry. Potential step voltammetry, in which current is measured after application of potential pulse. That means suddenly potential of working electrode is increased, and during that time the current is measured. Therefore, it is called as potential step voltammetry. Second type, that is called as cyclic voltammetry. Cyclic voltammetry. In cyclic voltammetry, the potential of working electrode is increased linearly to carry out oxidation or reduction of analyte. And then the potential is scanned in opposite direction, in reverse direction to carry out reduction or oxidation of analyte. Therefore, in this case, first of all, the analyte solution is oxidized or sometimes it is reduced. And in second step, the oxidized product or reduced product of that analyte is further converted to its original state by scanning the potential in opposite direction, and it is again obtained in its original form. And during the oxidation or reduction in first step, current is measured, and during reduction or oxidation in second step, current is measured. When magnitude of current in first scan is equal to the current measured in second scan, then the reaction is said to be reversible one. Therefore, basically, cyclic voltammetry is useful to study the reversible or irreversible reaction. Therefore, here second class of the potential program voltammetry is a cyclic voltammetry. Third technique or third type is called as stripping voltammetry. Stripping voltammetry is performed by means of two steps. In stripping voltammetry, first of all, analyte is concentrated onto working electrode by oxidation or reduction process. Or accumulation of analyte is carried out on working electrode by oxidation or reduction process, and the current is measured, by stripping or removing the analyte from working electrode. That means in this case, first of all, analyte is concentrated onto the working electrode either by oxidation or reduction process. Once that analyte is accumulated or concentrated on the working electrode, in second step, the analyte is initially oxidized, now it is suddenly reduced, or analyte which is initially reduced, which is suddenly oxidized, and then during this current is measured. Therefore, measured current is directly proportional to the concentration of analyte, and the potential at which analyte removes from the electrode is the identity of that material. Now, fourth technique or type of this voltammetric potential program voltammetry is called as amperometry. In amperometry, current is measured as a function of time. In amperometry, current is measured as a function of time at constant potential, and here generally the current measurement is carried out by adding some reagent into analyte solution, and that analyte is kept at a constant potential. Therefore, reaction of the analyte with the reagent is carried out at constant potential, and during this current is measured. Therefore, it is also called as amperometric titration. Therefore, amperometry, that is the fourth type of potential program voltammetry, in which current is measured as a function of time at constant potential. Basically, the analyte solution is kept at a constant potential. Another reagent is added into the analyte solution and current is measured. That is simply a process of titration, and during the titration, current is measured. Therefore, it is called as amperometric titration. Student, this is about the classification of voltammetric work. Student, with this, I invite you to my next lecture on polarographic analysis. Thank you.